So the question was, once the genome is sequenced, how is the data protected and who has access to it? So the answer to this depends on whether we're talking about clinical sequencing or research sequencing. Most of the time what happens in research setting, which is where genomes are being sequenced right now, is the samples are de-identified. So the patient's name is removed, their date of birth, and we give them a code. And that's how that genome is referenced. So it has, for example, it's called Genome 1, Genome 2, Genome 3. And then once the analysis is done, the sample is linked with the name by the group who is involved in the care of that patient. So essentially their doctor or their genetic counselor, those are the people who know whose genome it is. But as the data is being uploaded into more and more databases, there is the possibility that a person could be identified by looking at the sequence of their genome if somebody knows how to look for that. In terms of clinical sequencing, the people who do not need to know who this person is, so for example, the people who are running the experiment or doing the bioinformatics analysis, they don't need to know who this person is, so they would be given a code, but once the data comes back, it is identified. And so the idea is if this patient goes from hospital A to hospital B, then they can take that information with them.